I will speak about the preferred reporting items for systematic reviews and meta-analysis protocol 2015. I want to say that this will be a kind of summary of the information that we heard before or we will hear after this presentation. So let's go. What are the reviews or systematic reviews? Earlier, reviews showed the opinion of experts, but nowadays this completely changed. Because nowadays we mainly write systematic reviews, which are based on systematic literature search to find relevant articles in a topic. And they use explicit, systematic, and reproducible methods to synthesize the data. And they are usually supported by statistical analysis, and that's we call meta-analysis. With these reviews or meta-analysis, we can answer specific questions, research questions, and with this, we can draw conclusions or make decisions. And we have to say that systematic reviews and or meta-analysis usually provide a strong evidence. As I said before, meta-analysis is a statistical synthesis of relevant literature data. It can answer specific questions, and it shows also on which field we need more clinical trials or basic science studies to answer questions. There are two special types of meta-analysis, the so-called network meta-analysis, and about this we have heard a very nice presentation from Noemi. And there is another special type of meta-analysis, the so-called individual patient data meta-analysis. We can plan a meta-analysis to compare different treatment modalities, to compare diagnostic approaches, to test the sensitivity or specificity of different markers, and to compare prognosis. What is the PRISMA-P guideline? This is a guideline which helps us to prepare or plan a protocol for a systematic review or meta-analysis. And its aims are the uniformization of the reviews and to improve the quality, transparency, and accuracy of our work. It contains 17 items and 26 sub-items, which can be divided in 3 main sections: the administrative information section, introduction section, and method section. And these sections and points will guide us how to plan a correct review or meta-analysis. These points are harmonized with the PRISMA checklist. This PRISMA checklist is the publication protocol of a finished review or meta-analysis. It's different of the PRISMA-P because it contains as well some points how to publish our review and how to discuss our results. And this PRISMA-P checklist is harmonized as well with the Prospero. About Prospero, we will hear a presentation after mine from Judith Tang. Okay, let's start with the first section, the administrative information section. The first point in this section is the title, how to plan, how to find out a correct title. What is important, that the title should be as short as possible and as informative as possible, as Peter said before me. So more people can find our article and more citations we will get. It is important that we plan a protocol. We have to mention it in the title, that this is a protocol of a planned review or meta-analysis. Second point is registration. After we have a plan and we made a preliminary search in the databases, we can register our work or our plan in the Prospero registry. And after this registration, we get a number. And we should use this number in our article later and in every presentation that we make about this topic. Why is it important to register? Because it can reduce the applications and minimize the risk of bias. Because, for example, later, the readers can refer back to the protocol. And if there are discrepancies in the protocol and in the finished work, it means a bias. Okay, the third point is the authors. we should name every author. We should write down the name of every author, their affiliation, email address, and we should give the mailing address of the corresponding author. And we should describe the role or the contribution of each author. We can do later modifications in our protocol, but it's important that we have to mark it. What did we do? What modifications did we do? And why did we make that modifications? And these modifications as well have a chance of bias. We have to describe all financial or non-financial supports, the name of the sponsors or funders if we have some sponsors, and we have to describe their role in the review. And it's a potential risk of bias as well, because, for example, industry-supported meta-analysis or systematic reviews usually have worse methodological quality, and they have more favorable conclusions. That's why the quality of these works are worse. The second main section is the introduction section. First, we have to, as Peter mentioned, we have to write a clear and not so long background, what we know in the topic, and what can we add to the prior knowledge. So why do we write a review or a meta-analysis? We have to clearly describe our objectives. The easiest way to describe our aims is the PICO question. Andrea Parnitsky had a presentation about this. So the P is the patient or the problem, I is the intervention, C is the control group or comparator, and O is the outcome. The third section is the methods section. And within this, the first point is the RD eligibility criteria. So the in or exclusion criteria. We have to clearly describe this criteria because our search or the search for relevant articles will be based on this in or exclusion criteria. Okay, then we have to describe and write down what databases we used during the searching process. The three most popular databases are PubMed, Embase, and Cochrane Library. But as Zsolt mentioned, we have many other sources. For example, we can search in the reference list of other works. We can contact study authors to send us their work. We can use trial registries. And it's not recommended, but we can use gray literature. But these are usually low-quality works, and they are not the part of the commercial publishing or distribution channels. Then we have to describe our search strategy. How did we search in these different databases? And we have to detail the searching process for at least one major database. Why is it important? Because this will make our work reproducible. So if others do the same searching process with the same strategy, they can get the same results as we got. Then, if we finished the searching process, we have to manage our references and the articles. And for this, there are many softwares used. But we use, in the institute, we use EndNote to manage references and citations. And this helps us to reduce the data entry errors and biases. Then, if we have all the articles we need, we have to select the relevant articles in our topic. This should be done by two independent reviewers or authors. We have to write down the name, who did this process, and what did they do. For example, someone did the searching, someone did the title and abstract screening, someone did the full text screening. And if these authors have disagreements, then it has to be resolved, for example, by a consensus or by a third opinion. Why is it important? Because it can enhance the objectivity and prevents later mistakes. And if we finished this process and we have all the articles we need, then we can complete PRISMA flow diagram, as we can see on this slide. And we can write in the numbers of the different stages in the search strategy. After we have all the articles and we have this PRISMA flowchart, we have to collect or extract the data from the relevant articles. It's important that it has to be done as well by 2 independent reviewers. If they have disagreements, they have to resolve it by consensus or by third opinion. And usually, we collect data in an Excel file. It's important to contact a biostatistician before we start collecting data, because they can tell us how should we collect data, what should we collect, should we collect p-values or other parameters or no. So it's very useful to contact a biostatistician before we start extract data. It's important that in the protocol we have to list and define all outcomes what we will search. We have to describe all data we will search. For example, the PICO items, the duration and the frequency of the measures, the outcomes, what are the outcomes, what are the definitions of these outcomes. We have to mention every simplifications we will make later or assumptions. Why is it important? Because later the readers can refer back to our protocol, and if they find discrepancies in the published review or meta-analysis and the protocol, they can think that something is wrong here and it's a bias. For example, the selective reporting bias, I will detail it later on another slide. And if we want to add later a data to our protocol, we have to explain why did we add this data later. Okay, if we can make a prioritization in the outcomes, the most important outcomes are the primary outcomes, and there are secondary outcomes which are not so important, but we can collect data for them. We should use relevant and validated outcomes. The most frequently used outcomes are mortality and severity, for example. Then comes the risk of bias analysis. We have to write down in a protocol how we will deal with this bias, how will we evaluate the risk of bias, because it can influence the results. It's often difficult to use. There are many scoring systems, for example, the JDAS score, Newcastle-Ottawa score, Cochrane library tool, and others. Zsolt spoke about this detailed. And the risk of bias in the individual studies can influence our results as well. So we have to deal with it. After we have a complete Excel file and with the data, we give it to the statistician, and the statistician will perform an analysis if our results are homogeneous enough or not. If our results are homogeneous enough, we can do a quantitative synthesis of the data, so we can write a meta-analysis. If they are not, they are very heterogeneous, then we can do only a narrative review, a qualitative synthesis of the data. I want to speak now about some frequent biases. For example, the publication or dissemination bias. It means that studies with negative or insignificant outcome will not be published. This is the characteristic of small studies. That's why we call this small study effect. We can estimate this bias risk with the so-called funnel plot. And there is the selective reporting bias, which means that we report only, that we do not report outcomes we report outcomes due to their significance or direction or magnitude of the results. We have to deal with these biases. We have to write it down in the discussion part, and we have to deal with them. They are important because they influence the quality of our work. And the last point of this PRISMA-P guideline, that we have to describe the grade of the evidence we found, and we can do this with this grading system that Zsolt explained earlier. I collected here some important web pages where you can find these checklists, the flow diagram, and any other files which are important to plan a review or a meta-analysis. Thank you very much for the attention.